All right, continuing with our cell cycle, we ended up the notes we talked about when the cell gets ready to divide, that has to double its DNA. Okay, so this is actually two identical parts of DNA. This side and this side are the same. So before the cell got ready to divide, it only had the one. So it makes another side, and these are called sister chromatids. They're identical, and then the narrow part is called a centromere, and something's going to attach. If you watch the video, it's the spindle. It's going to attach, and it's just going to pull it apart. So this part is going to go to the new cell, and this part is going to go to another cell. So you're going to have two new cells from one, and they're going to have the same DNA. They're going to have the same genetic information. Okay, so during cell division, the two sister chromatids of the duplicated chromosomes, they're going to separate and move into two new nuclei. Okay, so when they separate, then now you're making sure that each new cell has the same amount of genetic information. Okay, once separated, the chromatids now they're going to become a chromosome. So the chromosome, when it's ready to divide, has the two parts of the chromatids. It looks like half a chromosome, but really each side is the same. And then when they go into the new cells, now they're going to spread out again, and then they're going to become a chromosome. Okay, so again, we have this is our chromosome. Now the cell's getting ready to divide, so it's going to make a copy of that chromosome. So now it looks like this. We have each part is the same, but they're called sister chromatids. Okay, and then when the cell divides, now each one gets, so now if you look, they're the same as, the cell they came from. So every time a cell gets ready to divide, the first thing it has to do is it has to double up its DNA, and then it splits it so each of them get the same. That's why every cell in your body has the same chromosome. Okay, so the cell division in a eukaryotic cell, now we're talking about true cells, we'll talk about prokaryotic cells later. But in a true cell, it's called mitosis. Okay, it's the division of the genetic material in the nucleus. And then when it gets all done, then you have cytokinesis, because you have to divide the cytoplasm and stuff too. So when we're talking about the beginning part with the chromosomes and all that, that's just what's going on in the nucleus. Okay, so that's going to divide, but we also have to worry about making sure each of them get cytoplasm too, and then organelles and all that other good stuff. Everybody has, each cell has to get the same amount of stuff. Okay, the gametes, again, those are the sex cells. They're produced by a different thing called, again, I call it meiosis. Some people call it meiosis. I don't know. You can call it whatever you want. Well, you got to either call it meiosis or meiosis, not anything you want. But anyway, that's something different. That's next chapter. Okay, but really what meiosis does, or meiosis, whichever we want to call it, you're going to end up with 4 cells, and they're going to have half the number of chromosomes. Because again, these are sex cells, so they're going to come together with another one. This, the egg has half the chromosomes, the sperm has half when they come together. Now you got the full number again. So that's something different that has to happen. But in mitosis, that's normal cell division. Each new cell has the same amount of stuff. From 1, you're going to get 2, and each new cell has the same amount of genetic material. Okay, so the mitotic phase, okay, it alternates. They have a phase called interphase. And when I was going to school, they used to call it the resting phase, which is not true because there's no resting going on. What's going on during the interphase is this is like the normal cell, when it's producing hormones or whatever that cell's supposed to be doing. That's what's going on there. So Walter Fleming, he's the one, he came up with a series of dyes of ways of looking at cells so you could see the chromosomes so that you got a better idea what happens during mitosis. Okay, so the cell cycle, again, that's what this chapter is about. First, okay, we're going to talk about mitosis. All right, this is the cell division. And then after the dividing of the chromosomes, that's mitosis. Then we also have cytokinesis. And cytokinesis is splitting up the fluid inside and making sure everybody, each new cell gets the same amount, plus the organelles and all that good stuff. Okay, and then in between, when the cell isn't dividing, it's in interphase. Like I said, when I was in school, they used to call it the resting phase. And there's like, no way, the cell's not resting. It's just the in-between when it's not dividing. Okay, this is when it's doing its normal job, what it's supposed to be doing. Okay, so it's doing its normal job, and then it gets to a certain size, and it's like, oh, time to divide. Then it divides. Okay, but when it's not dividing, it's in what they call interphase. Okay, interphase is about 90% of the time. And that's when they're doing their job. Okay, and it can be divided into, they have 3 different phases, what they call 3 different parts of interphase. Okay, G1 phase, that's when it first gets done dividing. It's its first gap. Okay, now synthesis is now when you're doubling up, you're getting the DNA ready, you're making sure that you got everything double, so if when it does divide, it gets it. And then they have the second gap. I think I have a better one. Oh, the cell grows during all these phases, but the only time it's duplicating the chromosomes is in this phase. So this happens when it first divides, and it's, growing and getting things ready. Maybe it's making some proteins or whatever it's supposed to be doing. Then during this phase, during the synthesis phase, remember synthesis means to make something, it's going to double up its DNA, its genetic material. And then the second gap is it's still growing and it's getting ready, but it's not dividing. Okay? And again, you can see in a cell cycle, okay, this is when it first divides, and it spends quite a bit of time there, almost, probably about 40% of the time in here. Then we get into this phase, okay? This is when we're making more DNA because it's going to have to divide, okay? And then this phase, and you can see this is the biggest phase, and then this one, and you can see mitosis and cytokinesis are only about 10% of the cell cycle, okay? So 90% of the time the cell's in interphase, and then it has, this is like going on with its normal life here, doing the things it's supposed to be doing. This is making copies of the chromosomes, so when it divides, it's ready. And this is, finishing up, still working, still doing its job, but getting ready to divide. And then the actual division, the mitosis, this is the division basically of the genetic material, the nucleus. And then the cytokinesis, the fluid, the cytoplasm, and all that. Those two really don't take up a lot, about 10% of the cell's life cycle. Okay, here's some more pictures with some dyes so you can see them better here. And honestly, it'd be really hard to tell the difference between G2, G1, and synthesis in a cell. They kind of all look like this. So if you saw a cell like this, it could be in G1, it could be in S, or it could be in G2. All right. So, but now what happens now to tell the difference between the phases, you're going to start seeing the chromosomes. Okay. And then things are going to happen. And these, I'm going to go through them first. But there's phases in mitosis, which is crazy because there's 3 phases in interphase, and that's 90%. There's 4 phases in mitosis, and that's probably only 5% of the cell cycle. Okay, and here's metaphase, anaphase, and telophase. But it shows you the pictures of them. You can go back through these. Let's talk about the phases. And again, this is interphase. The cell just looks like it would under the microscope. We've got, really, when you look at a cell under a microscope, this, the standard light microscope, you can see basically the nucleus and the cytoplasm and the outer membrane. You can't really see a lot of the other stuff in there, okay? Which is crazy because when a cell gets ready to divide, you can actually see the chromosomes. You can't hear there. You can see the DNA is all spread out. It's really thin and here, but now it starts to coil down like I talked about before, and now you're starting to see those chromosomes. And then the chromosomes line up, okay? They have, okay, when you first see the chromosomes, that's called prophase. And then, yeah, as you can see, they're starting to get ready. They're not lined up yet, but so this is prophase. And then they line up in the middle. That's metaphase. So when they're in the middle, that's metaphase, okay? And then they start to split, okay? Each one of these, remember, these chromatids, they're going to go to opposite ends. That's anaphase, okay? And then when the cell starts to actually form like what they call a furrow, this is where it's going to break into 2. That's telophase. And then right after telophase, or during telophase, because it's splitting, you're going to get that splitting of the cytoplasm and everything else too. So these 2 phases kind of occur together, okay? So mitosis is prophase, metaphase, anaphase, telophase, okay? We used to remember, like I said, the 4 phases. Our saying was, and I don't know why we use Princeton, because maybe we thought we were Ivy League or something, but Princeton made a touchdown. Prophase, metaphase, anaphase, telophase. You can do anything. You can put panthers, you can put, I don't know, poopy heads, whatever you want to call. But it's prophase, metaphase, anaphase, telophase. And those are the phases of mitosis. And basically, you can see them using a regular light microscope. It's kind of cool, actually. Okay. Oh, here's a little video. I know I don't usually show you videos, but watch this cell. You can see, now you can see the chromosomes. They're all visible, which you couldn't before. Now they're getting all lined up. Okay, once they're all lined up, it's in metaphase. Okay, and then they get like little spindles attached. The spindles you can't see under a regular microscope, you guys. And now, once they get lined up, then they split. Each sister chromatid go into opposite sides. Come on, guys, go to opposite sides. I know it's a short video. And so there you go. See, they split. Now they're in anaphase. And then it's going to keep going and it's going to split into 2 cells. That's telophase. And now you have 2 new cells. Okay? All right. So a closer look at that little spindle. And you cannot see spindles when you're looking under a light microscope. Okay, they're microtubules and they're really, really, really thin, so you can't see them. But they actually control the movement. There's kind of a debate whether it actually pulls them apart. Most people say it's just like a path for the chromosome to follow. So I don't even know if they actually pull them apart. But the spindle attaches to the centromere on each chromosome. And then when they split, it gives them a path to follow. Okay. In animal cells, there's an extra thing on the ends. On the 2 poles, you'll see a little dot. And that's called a centrosome. And that's where all the spindles attach to. You don't see it in plant cells. Not sure why. Maybe plants said, hey, we got cell walls, we got chloroplasts, so we don't need any more organelles. We'll pass. I don't know. But animal cells have this centrosome, it's called, and it's just a spot on each pole, and then all the spindle fibers attached to it. Okay. The centrosome replicates during interphase as well. It forms 2 centrosomes, one on each end, and they migrate to opposite ends of the cell, and that happens during prophase. They call it pro-metaphase. It's kind of the beginning. You guys, we're just going to go with prophase, metaphase, anaphase, telophase. We don't need to do in between those. We got enough to learn, right? But anyway, that centrosome, which is on opposite ends of each other, and the spindles attached to it, they migrate to opposite ends, getting ready to divide during that interphase. Okay, and then they call it an aster. That's where you got the spreading out of from the centrosome, you got all those spindles spreading out. They call that an aster. It looks like a radio ray or a star. Okay, the spindles include the centrosome, the spindle microtubules, and the asters. Okay, so that whole thing from the dot, and then all the fibers that are attached. Okay, those are called the spindle. Okay. And here they have during pro-metaphase. Again, this is a phase kind of in between prophase and metaphase. This is where things are starting to get lined up. But some of the spindle microtubules attach what they call to a kinetochore of a chromosome and begin to move them. Okay. Again, it's pro meaning before metaphase. We're just going to stick with prophase, metaphase, anaphase, telophase. But you'll see these and you'll see the word come up. So I didn't want to leave it totally out and confuse you. Okay, kinetochores are protein complexes. And again, they help with those centromeres, the middle part, okay, of the chromosome, where the 2 chromatids attach, there's a centromere. Some of them, they have this protein complex on it. It gives it a little extra handle to hang on to, I guess. Okay, so at metaphase, the chromosomes are all lined up. It's easy to remember metaphase because they're in the middle, metaphase. Okay, they're all lined up on that plate in the middle, and they're in between the 2 spindle poles. Okay, and then the spindle fibers are going to be attached to the middle, to the centromeres, and then when it gets to anaphase, then they're just going to pull apart. Okay, so here you can see the spindle fibers. Here you can see the chromosomes. It's nice when we draw it out. They're all just nice, perfectly lined up. But when it really happens, you can see they're kind of scattered, but they're in the middle. So this is in metaphase, and it's getting ready. They're lined up. They're getting ready to split. And then here's those centrosomes. They're kind of a gathering where all the spindle fibers attach, okay? And really a spindle fiber is a microtubule. We talked about those in organelles, okay? In anaphase, the cohesions, those little connections, they are broken up by an enzyme. There's an enzyme called separase. Well, it separates them. That's kind of cool. And then the sister chromatids will separate and move to opposite ends. So they actually have like an enzyme that breaks them down. It's an X, and it breaks it down into the 2 parts. So it goes down through it and breaks it into 2 parts. Okay. And that's called separase, which separates them. Okay. So now they're going to move to opposite ends of the cell. So anaphase is when the chromosomes break into 2 and start moving to the other sides. And then the microtubules help by shortening that. They're the ones that kind of pull them apart. Okay. And I hope that's the last word. Yeah. Oh, we'll end up here. You can see it right here. We're out of time. So this is what we'll pick up next time. All right.